fall day in Ann Arbor. I think it's a wonderful warm day in Palo Alto, California as well. Dr. Gwilliams is assistant professor at Stanford University, where she's been since 2023 in the departments of psychology. She's co-director of the Center for Neural Data Science and faculty director of the Core Human Neurosciences Community Laboratory. Dr. Gwilliams completed her PhD in 2020 at New York University, working with Alec Morantz and David Popel, followed by postdoctoral work at University of California, San Francisco, in Eddie Chang's lab. So if you want to understand where neurolinguistics is, and more importantly, where it's going, you look to Dr. Gwilliams' work. That's where the field ought to be, and I think will be going, from single neuron recording of speech perception to spatial and temporal dynamics of phoneme, morpheme, and sentence processing. The work identifies, kind of paves the way towards a real qualitative shift in how we can understand the connections between linguistic representations and computations on the one hand, and rich, complicated, high-dimensional neural systems on the other. At the heart of Dr. Gwilliams' work, at least in my view, is a focus on interfaces. So interfaces from phonetics to phonology, from morphology to syntax at the linguistic level, and interfaces between that rich, sometimes noisy neural data and symbolic and explainable theories of processing and representation. Throughout the work, one sees payoff when attention and careful, rigorous thought is brought to bear on the junctions between each of these things, and that's where insight comes from. So the work appears in venues like Proceedings from the National Academy of Sciences, Neuron, Journal of Neuroscience, annual reviews, Nature. That's this year, okay? The work is funded by a range of institutions and philanthropic foundations. This includes early career awards from the Whitehall Foundation. Dr. Gwilliams is also a recipient of awards from the Cognitive Science Society and the Society for the Neurobiology of Language, among others. I'm just absolutely thrilled, Laura, that you're here today to share your insights with us. So please join me in welcoming Dr. Gwilliams. Thank you.
Thank you so much. That is the kindest introduction I've ever received, John. Thank you. I'm actually tearing up a little bit. So yeah, thank you. I hope that I can do myself justice with my talk today. So let me just share this here. Great. So do you see what you think you should be seeing right now?
Yeah.
Give me a thumbs up. Yeah. Okay, great. So thank you so much for inviting me to visit today. I've had some really fun meetings with trainees and faculty earlier today, and I'm really delighted to get the chance to also present my research program and what it is that I'm trying to achieve, and some of the progress that we've made to answering some big questions and understanding speech neuroscience. So I'll get right to it. The big question that my lab tries to answer is, how does the human brain take the sensory auditory signal of speech and transform that into meaning? And this is like, we do this all the time automatically without even thinking about it. And yet it is not a trivial problem to solve, transforming arbitrary sounds into very complex, rich, and often meanings that you've never heard before. So I'm trying to understand this processing system that allows all of this to happen. And this is a pretty big question to try and get a handle on. So to make sure that I'm making progress towards answering this question, I actually try to subdivide it into a series of smaller questions. So one sub-question is, okay, we're going from sound to meaning, that's input to output. What are the neural representations that are generated in order to form this mapping? So, to be concrete, examples of representations, it might be the cochlear representation of sounds, or it might be the phonetic features in speech, or it might be the syllables or morphemes that you're hearing. We're trying to understand what are the fundamental units and format of information that the brain is generating when processing speech. The second sub-question is what operations are at play? So how do we derive one representation from another? How are these representations actually manipulated? So some examples of operations might be filtering of the input signal within a certain frequency range, or it might be performing some kind of non-linear categorization. So maybe you want to go from an acoustic representation of a sound to something more discrete, like the person said an N sound and not a B sound. And then finally, we need to understand the order in which all of these processes happen. So in what order are representations created? In what order are operations applied? And we can kind of summarize that question as information flow. So my work is trying to address each of these sub-questions in understanding speech comprehension. And one important way in which I do this is by actually working with neural recordings that operate at distinct spatial scales. So some of my work uses non-invasive whole cortex recordings. This would be something like EEG and MEG. Some of my work uses intracranial EEG. So this is electrodes that are implanted when someone needs surgery for epilepsy. And then at the most fine-grained spatial scale, we now have technologies that enable us to record from single neurons to see what those single neurons are responding to about the speech signal. And I think it's really important to understand speech processing at each of these spatial scales, because ultimately speech comprehension operates over all of those scales. It's not just about what the single neurons are doing, it's also about how whole cortical networks are communicating. And I'm trying to piece all of these pieces together. Okay, so that's quick snapshot of the type of questions I'm interested in and the types of approaches that I'm taking. And now I'm going to go over three studies that work with two of these modalities. So I'll first talk about two studies that used whole brain MEG, and then I'll close us out with a study that recorded single cells with neuropixels. Okay, so starting off, in this work, I was primarily interested in representation. So that first sub-question that I introduced. And I'm particularly interested in representations across the hierarchy from sound to meaning. So if we go back to this slide, I said that one of the fundamental challenges that the brain needs to solve is mapping from sound to meaning. And it's believed that the way that the brain does this is by generating a hierarchical set of representations that, as you kind of move along this hierarchy, the representation becomes slightly less like the acoustics and slightly more like the meaning that's trying to be derived. And so one fundamental question is, okay, what are the representations that form this hierarchical bridge? The second question is, what order do these representations actually get generated by the brain? I may be like, okay, Laura, you had me with the first question, but the second one is a cheap shot. Like, obviously, things go from phonemes, you assemble those to make syllables, those to morphemes, etc., right? You're just going to, in a bottom-up fashion, assemble smaller items to create larger and larger ones. However, if you're listening to continuous speech, you might actually, because of the ability to anticipate what someone is about to say, actually have a sense of what someone is trying to say at the meaning level before the person has actually even finished saying all of the phonemes and syllables in their utterance. So if you can anticipate what someone is going to say, you might actually have a representation of the higher order meaning before you have a representation of the lower ones. So maybe you know the meaning of what someone is going to say, but you don't yet know exactly which word out of, say, 4 possibilities they're actually going to choose to use. So that sets this up as a non-trivial question when listening to continuous speech. This non-trivial question becomes, are things indeed assembled in a bottom-up fashion? Or do we actually see evidence for a reverse hierarchy because of the fact that people can predict what's coming up? Okay, so those are our 2 questions. And we address this question by inviting 21 native English participants to come and have their brain scanned with MEG. This is an MEG system, while they listened to 2 hours of audiobooks. And as they're there listening to these audiobooks, we interrupt the book every 4 minutes or so with a simple comprehension question. So it might be, what state was the bank that the robbers were robbing? And that's just to make sure that people are paying attention and that we have an estimate of their behavioral comprehension. But everyone was at ceiling in doing this task, so it mainly just served as a attention task. Okay, so we have people listening for 2 hours, and that corresponds to neural responses to about 40,000 phonemes per person and 16,000 words, which is one of the great benefits of using this type of naturalistic paradigm where you're just asking people to listen to continuous speech, is that every single second of that recording is data that is usable. There's no inter-stimulus interval, which is just kind of like a buffer time. Like every single moment is usable. And so you end up getting a lot of usable trials by using this type of paradigm. And we actually realized that this was one of the biggest data sets of its kind when we recorded this. So we decided to organize it nicely and release it publicly. So if you have a need for analyzing naturalistic speech listening data set, then please, I urge you to make use of that. Okay, so we have people listening to these stories, and we annotated those stories for a rich set of hierarchical features, like I introduced earlier. So we annotate the stories for the phonetic features of each sound in the words, properties relating to the sublexical structure, like how many syllables and morphemes are in each word, properties of word class, so is this a determiner, adjective, noun, verb, and then features relating to the actual syntactic structure, like how many branches of the tree can we create at a particular word, how many can we merge, and how deep into the tree structure does a particular word occur. And then finally, we have proxies for the actual meanings of the words and phrases, which we make use of simple large language models to extract word embeddings to be able to represent higher order meaning. So I'm just showing you a subset of the features here. We actually had about 54 in total, but we organized them into these 6 feature families that span this hierarchy. So one question, as I said, is which of these features seem to be processed by the brain? But another important question is in what order are those representations actually generated? So to be very clear what the hypothesis space is here, I'll give you the 2 extremes of the hypothesis space. One is this bottom-up hypothesis, which, as I introduced, says, okay, obviously you're going to process the phonetic features and then those will get assembled to make syllables. So that's when we'll be able to decode the sublexical stuff and so on and so forth. So if processing is unfolding in this bottom-up way, we should be able to decode the lower level properties of speech earlier than we can decode the higher level properties. The alternative is this reverse hierarchical idea, which says, but hang on a second, people are listening to full-length stories here. They're going to be able to anticipate the semantics more than they can anticipate the specific sounds that someone is going to say. And so actually, we will be able to read out the higher level information of speech earlier than we can read out the lower level information. Okay, so I'm going to show you the results of our decoding analysis on the right-hand side here for each of these 6 feature families. So let me kick us off. And on the x-axis here, we have time relative to word offset, and we're fitting our decoder at each millisecond in time, so at different lags relative to when each word ended. And so that's what is going to give us these time-resolved decoding curves. So yeah, if I start us off with the decoding of phonetic features, you can see there's multiple lines in this plot. That's because there are multiple features in this feature family of phonetics. So like one of these lines is nasals and one of them is fricatives, etc. And this bar here is the result of a temporal permutation cluster test. So it tells you when in time can we decode this feature family better than chance. So we can read out the phonetic features from about 0 to 200 milliseconds. The sublexical features are about -100 to about half a second. Word class about -200 to 200 milliseconds. Syntactic operation for about -200 to about a second. And then syntactic state and the semantic vectors were decodable for about half a second before the word ended until about a second and a half afterwards. So one thing we can already tell here is that all of these features that we're trying to decode from neural activity, which I selected based on essentially, has any linguistic theory ever suggested that this might be something that is important for processing language? We put this in our annotation scheme. All of these features do indeed seem to be encoded in brain activity, thus we're able to decode them back out again. So that's exciting because it suggests that these linguistic theories, which are really good at describing language structure, are also really good at explaining neural responses to that language structure. The second thing I want to point out is the really interesting dynamics here. So we indeed see that the higher level features seem to be encoded earlier than the lower level ones. And you might also notice that the high level features seem to be encoded for a longer period of time as well. And something else I like to point out is just how much parallel processing is going on here. I really just think this is a beautiful demonstration that we can take this 200 millisecond chunk in time and you can simultaneously read out like that the word ended with a fricative, that it had this many syllables, that it was a noun, that the tree structure was about this complicated, and it had something to do with dogs. All from just one snapshot of brain activity. So that really goes to demonstrate just how much parallel processing is happening of all of these different hierarchical levels at the same time. So if I pull back those 2 hypotheses that I introduced, the hypothesis that predicted this reverse hierarchy seems to be the best fit to our data. And it's really interesting that this is the hypothesis to win out here, because this hypothesis has been primarily put forward for visual processing and way less for auditory processing, kind of even less again for speech and language processing. So what this has to say for itself in the visual domain is, well, the information that's hitting your retina is actually really noisy and ambiguous as to the visual objects that you're trying to recognize. And so you actually use the context of, say, other objects that you've recognized recently to help and guide and bias how that low-level visual information is actually processed and interpreted. And essentially, I think the same thing is happening here for the case of speech processing, that you have the information at the higher levels, which tells you, okay, this person is talking about their dog again, which helps to guide and bias how the low-level information is processed and interpreted, given that it's much more vulnerable to noise and ambiguities. So if I summarize all of this together, what I think is going on is that when you're listening to continuous speech, you have the ability to make predictions about what's coming next. And so we have this kind of top-down pass of information, which is a predictive pass. And then when you actually get the bottom-up input, you do a subsequent compositional integration pass over the information. And that's important because obviously your predictions are not always going to be correct, right? So I could stop talking right now and you could just hallucinate the rest of this talk for me if your predictive system was perfect. But obviously that's not the case. Sometimes you predict the wrong thing. And so it's important to also be able to update your interpretations based on what actually happened. But in the cases where you predicted the right thing, that's really beneficial because it means that you can process things more robustly and faster. And then on top of this two-pass, as I mentioned, we also have this really high degree of parallel processing, which I think is a big part of what allows these different levels of representation to actually interact with one another and to disambiguate the information at one level based on the information that has been derived from another one. Okay, so this is the work that we've done on hierarchical processing so far. I did want to give you a little flavor of one of the projects that we have ongoing now, where we're actually trying to investigate the developmental trajectory of this hierarchy. So we're working with a really unique data set of intracranial EEG from 41 children, while they also listen to similar audiobooks. And we're trying to understand at different ages, what does the hierarchical process look like? So do we find that, for example, there's more higher order predictive processing for younger children, or maybe that doesn't kick in until later? There's some really interesting ways that this could come out, and I'm really excited to figure out how the mature system that I just presented to you actually comes to be through development. Okay, so now I want to talk about a second study where we were more interested in the operation side of this question. So here we wanted to understand how the brain actually sequences rapidly unfolding phonemes during speech processing. And so relative to this hierarchy that we just talked about, I'm now going to focus on these lower two levels. So looking at phoneme sequences and how we think that might get assembled into syllables and slightly higher order structures. That John also nicely introduced, kind of looking at the interface of how you transition from one level of representation to the next. So how this study came to be actually was based on an observation that I made in previous work, which is if you ask people to listen to just isolated words now, this not continuous speech, and let's say you have a word, you have word pairs that either begin with P or begin with B. What we found is that you can read out whether the sound was P or B from the neural activity for a really long period of time. So the duration of this sound itself, like in the sensory input, is only about 100 milliseconds, but you can carry on actually reading that out from the brain activity for about 4 or 5 times longer than that. And the point of this previous study was to actually look at the role that plays for disambiguation. So what I just described is a really good processing strategy if what you're trying to do is make use of information in the future to disambiguate what happened in the past. So let's say our word pair was parakeet. and barricade. But if you weren't quite sure what that first sound was, let's say it was ambiguous between P and B, well, if you're patient enough to wait to see how the word identity resolves, the word identity also disambiguates the identity of the sound that you heard earlier. And so having this longevity of information processing is what I think actually enables future information to influence the perception of the past. But it still leaves this kind of conundrum that basically we're saying that, okay, the phoneme itself lasts 100 milliseconds. Let's say you're hearing a really simple word like hello. What I'm saying is that the first sound of this word, huh, you hear that and it's processed, let's say, for 400 milliseconds in this brain area of superior temporal gyrus. And then obviously before you finish processing this first sound, you're already hearing the second one, and it's already being processed in the same brain area of superior temporal gyrus, and so on for the third and for the fourth sound. So by the time you're hearing this fourth sound of this word, your brain is still processing the previous sounds in the same brain areas. So this raised two questions that I wanted to address here. First is, hang on, how does the brain not get confused? If it's trying to process all of these different sounds in the same place, how does it not get confused about what sound belongs where or the identity of those sounds? And second, how does it correctly recall the order that the sounds came in? Because that's obviously going to be critical for figuring out what word it is that somebody is saying. So these are the two questions that we wanted to address here. And we addressed it using exactly the same data set that I already described to you. So this is 2 hours of MEG recordings, people listening to audiobooks, but now we're going to really focus in on the processing of each of the phonemes rather than looking at processing of words like I showed you in the previous work. Okay, so we have these continuous stories that people are listening to, and we still need to do annotations, but now we're going to critically annotate when each phoneme happened in the speech sequence, as well as what the phoneme was, what its phonetic features are as well. And this is going to allow us to decode from the neural activity what the phonetic feature was that the person was hearing. But critically, we're going to do this as a function of the sequence. So we're going to decode the phonetic features for the first phoneme of the word, and then separately the second phoneme of the word, and separately the third phoneme of the word, etc. So we can get a handle on how these sequences are being processed and assembled. Okay. So the specifics of the analysis goes a little bit like this. If you're familiar with temporal generalization analysis, we're essentially doing that with a slight tweak. If you're not familiar with that, then don't worry. Hopefully after this explanation, it'll all become clear. So we're training our decoders for each of these features separately. So let's say we have a decoder that is classifying fricative versus not fricative. And we're going to train that classifier at a separate classifier for each moment in time relative to when that sound began. So we'll have a classifier at 0 milliseconds that uses the MEG sensors to say this was fricative or not fricative, and then a separate one at 100 milliseconds and a separate one again at 200 milliseconds. Why do we do that? We do that to enable the classifier to find what is the pattern of activity over the MEG sensors at that moment in time to read out this information, because the information itself might not be static over time. Then we evaluate the accuracy of all of these classifiers, again at different lags, but now we're doing that relative to when the word began. So we'll evaluate the accuracy of our classifier at the beginning of the word, and then later and later in the word. Okay, so yeah, so we're training on all of the phonemes at different milliseconds relative to phoneme onset, and then we're evaluating the accuracy of those classifiers relative to their position in the word. Okay, so if that's still a bit fuzzy, I think it becomes easier to understand when I actually start showing you data and interpreting it. So let's start off with decoding the phonetic features of the first phoneme of the word. That's what I'm showing here. So this is a contour on the decoding performance. And the way to read this is to say, okay, starting at 100 milliseconds after the speech sound began, that's when we start to be able to decode this, the phonetic features of the first phoneme of the word better than chance. And then we continue to be able to read that information until about 450 milliseconds. So the overall duration with which we can decode this information lasts for a long time, just as I introduced to you. What's important here, though, is that if we look at any horizontal slice here, we actually find that the width of this horizontal only lasts at about 80 milliseconds. So what that means is that the neural pattern that is informative at, say, 100 milliseconds is different from the informative pattern at 200. And it's different again at 300, and it's different again at 400. So all of those patterns are informative. All of the neural responses contain this information, but which neural responses are actually containing that information is changing over time. So this leads us to conclude that the neural representations are actually evolving as a function of time. And we see evidence for this dynamic code, not just for the first phoneme of the word, but also for the second, the third, the fourth, the fifth, as well as the last phoneme of the previous word, working backwards as well. So all phoneme positions seem to share this processing strategy that they use a dynamic code to encode the information. And we realize that this actually equips the brain with a number of processing advantages. So if I just highlight a horizontal slice of this plot here, so this is at 200 milliseconds after these sounds begun, you see that you can, there's very little overlap between the neighboring speech sounds. So the contour of one phoneme has very little overlap with its neighbors. Why is that? It's because by the time the next sound comes in, the previous sound has shifted its neural pattern, so they're not trying to occupy the same pattern at the same time. And I think that this is exactly what stops the brain from getting confused about processing multiple sounds at the same time, because it's never trying to process it using the same neural pattern at the same time. And so this lack of representational overlap, I think, is what solves this problem of juggling, processing lots of things at once. So if I also take a vertical slice here, let's say at half a second after, like half a second into the word, you see that we can indeed read out the history of 3 previous phonemes. So this again confirms our intuition that if you query the neural response, you actually can read out a history of multiple sounds at the same time. The thing that I find maybe most exciting about all of this actually is that not only can you read out the history of multiple sounds like in parallel, you can also read out the relative order with which those sounds actually entered the ear by virtue of what neural pattern it is that's encoding that information at the point that you're querying it. So you know that more time has gone by since the first sound began as compared to the second, as compared to the third, and therefore you know the order that those sounds actually came in. And so you can use that to assemble this kind of ordered trigram into a higher order structure, such as syllables and morphemes. Okay. So here is the same data, but just with a slightly different visualization. where this was 2 hours of listening with MEG. And then my postdoc, Jill Crees, was interested to assess, okay, if we only had 30 minutes and we're recording with EEG, can we still uncover these phoneme sequences? And in addition, she was interested in actually looking at whether phoneme sequence processing might be affected in people with aphasia, and specifically people who have phonetic processing difficulties because of their aphasia. So in Jill's work, she ran exactly the same analysis that I described, but now based on 25 minutes of listening with EEG with much older adults. So we have 70-year-olds and without aphasia, and then an average of 70-year-olds with aphasia too. And we find that this analysis approach is it is certainly noisier for less data and with EEG, but that we can still read out these sequences from the neural activity, and we see some interesting differences between the control participants and the individuals with aphasia. This is a preprint. I know that I talked about this work with a couple of people in the meetings earlier today. So if you wanted to read about the details of pre-processing or analysis approach or anything, it is all described in the preprint. Okay, great. So one final point along these lines. So I just told you that we see this dynamic code for the phonetic features. And this is the same data, but now instead of separating it out for each phoneme position, I've just collapsed them all together into one plot. Okay, so this diagonal shape suggests that we have this dynamic code. Maybe the next natural question to ask is, okay, is this a special phonetic thing? Or if you look at other levels of representation across the hierarchy, do you see evidence for a similar type of dynamic? So we reran this analysis, not just at the phonetic level, but also sublexical, lexical, syntactic operation, semantics, and syntactic state. And essentially, across the full hierarchy, we see evidence for this dynamic code. And you might also appreciate, and if I collapse these together, you maybe see it a bit clearer, that the actual width of this diagonal changes as a function of where you are in the hierarchy. So the width is much narrower for phonetics, a little bit wider for lexical features, and most wide for the syntactic state features. And the width is what tells you how quickly the neural code itself is actually evolving. So it seems that the code for phonetics is evolving much faster than the code for words and phrasal information. So the speed of this evolution seems to scale with unit size. And why I think that is that if the kind of purpose, or one of the purposes of this dynamic code is to avoid representational overlap between neighbors, well, neighbors are much closer together if you're at the phoneme level, and they're further apart if you're at the word level. And so it seems that maybe the code is actually evolving at exactly the pace it needs to evolve in order to avoid overlap with its neighbor. And we're following up on this work now, so I don't have any plots to show you, but actually manipulating things like speech rate to see whether the speed of this process actually scales with the speed of the input, or whether the neural dynamics are inherent and there are limits on the speed that they can unfold. Okay, so our final study that I want to talk about is now zooming in to processing in single neurons, where essentially I want to understand the neural implementation of some of the processes that I've talked about so far. So again, to introduce this, up until this point, we've been looking at whole cortex MEG activity, and now we're going to go all the way to the other extreme, looking at responses in single cells using these neuropixel probes. So just a little bit of background to motivate this. I've mentioned superior temporal gyrus, which is this gyrus here, that has been implicated a ton in processing speech. So it seems that different neural populations in this brain area encode phonetic features, as well as other types of speech features like fluctuations in the envelope and fluctuations in pitch. So this brain area seems to be critical and really involved in a lot of speech processing. And we understand quite a bit about how these processes are actually kind of spatially organized over the gyrus, thanks to intracranial recordings with electrocorticography, for example. So you kind of find along the surface, there's like a salt and pepper kind of smattering that you'll have, okay, this neural population really likes plosive sounds, and maybe next door to it, it really likes fricative sounds, etc. Okay, so we know quite a bit about how this area processes speech features from like a surface perspective, but what we don't know anything about is how this processing actually is given rise to based on what the single neurons are doing in that area. So at any one surface location, there is a layered structure. There are 6 layers in the temporal lobe, which at different depths relative to the surface, they have different types of neurons living in there. There are different amounts of recurrent connections. And this has been studied a lot in animals of how neurons that live in these different layers relative to the cortical surface seem to be involved in slightly different computations. But when it comes to speech processing in humans, we don't know anything about this at all. So that's where this work comes in. So we wanted to ask, okay, if we can record not just from the surface of the brain, but actually record from neurons at different depths relative to the surface, what do we find? Do we find that all of those neurons are tuned to the same thing? So in this case, are all of these neurons encoding fricative sounds? Or do we see that actually different neurons, maybe organized by different depths, encode different types of speech features, and it's not all encoding the same thing? So this leads us to 2 specific questions. One is, if we're able to record single neurons in the superior temporal gyrus, what do we find those single neurons actually are tuned to? And second, how is that tuning organized as a function of cortical depth into these 6 cortical layers? Okay. So before we get to those 2 questions, first I need to explain a little bit more about how you actually record neurons across the cortical depth. And that's where these neuropixel probes come in. So these are a huge leap forward in nanotechnologies of being able to, so this probe, it's a probe which is 1 centimeter long and 70 microns wide, so very, very, very thin. And along this 1 centimeter length are packed with 400 electrode contacts that you can record from. And so that gives you the ability to record hundreds of neurons at the same time at different depths along this probe. And for context, before this probe was invented, when people wanted to record single neurons, they would be able to record maybe like a dozen at a time. So the ability to record hundreds at the same time is a huge advance. And this has really propelled neuroscience research with animal models for being able to understand single neuron processing across cortical layers. And a big part of our job was figuring out how do you safely translate the use of neuropixels probes for recording in humans. And the way that we do this is by working with patients who are having brain surgery, either to treat epilepsy or to treat a brain tumor. And we insert this neuropixels probe into tissue, which is about to be resected as part of their clinical care. And so, yeah. And yeah, this is very complicated and took a long time to determine how to do this effectively and safely. So we recorded with 8 patients with insertion sites spread along the superior temporal gyrus. And the exact insertion site depends on the surgical plan of that given patient. So here are the different insertion sites, and then we can see here the MRI of the first participant with a schematic of the probe insertion for that participant. So you can see that we insert the probe so that there are some contacts in white matter and some contacts in air, so that we're certain that we're spanning all of those cortical layers in the gray matter. And then because the tissue is being resected anyway, actually allows us to perform histology Nissl staining so that we can relate the electrophysiology with the particular layer that we're recording from. Okay, so I want to show you some of the data that we get from this, and I need to share my share computer sound. Okay, so participants were listening to isolated sentences. We took these from the Timmett corpus, if that means anything to you. And for some of these sentences, we actually repeated them a number of times so that we could get a sense of the reliability of firing across repeats. So for this sentence, it was nobody's fault. Here's the phoneme transcription, the waveform, the spectrogram. And I'm going to highlight here the nasal sound because that might become relevant in a moment. And then we can also time lock the spiking of neurons in time with the stimulus being presented. So here, the time axis corresponds to the time axis of the stimulus above. So each red line is one spike of this one neuron, and each row is a repetition of exactly the same stimulus coming in. And I'm going to play this movie now where you should see the remaining 5 trials get filled in, and hopefully you'll be able to hear the stimulus along with the spiking.
It was nobody's fault. It was nobody's fault. It was nobody's fault. It was nobody's fault.
Did you guys hear that? Yeah, great. Okay, perfect. So what I hope you appreciate here is that this neuron fires at exactly the same time, roughly the same number of spikes every single time the same sentence is played. So we can now look at the responses of the same neuron, but to a different sentence. Junior, what on earth's the matter with you? Let's do that.
Junior, what on earth's the matter with you? Junior, what on earth's the matter with you? Junior, what on earth's the matter with you?
Okay, so what we find is this same neuron is still responding very reliably every time this different sentence is played and fires at the same time every time there's a repeat. But its timing is different from what it was from the first sentence. And this neuron, as I'll show you in a moment, actually seems to be responding to when nasal sounds occur in these sentences. So we see this reliability of spiking, not just for this one neuron that I picked, but we can do this for different neurons. And I'm showing in the different rows for different sentences. And you can see that there's very reliable timing of the spikes for these repeated sentences, suggesting that these neurons are encoding properties of the speech that they're hearing. So the way that we statistically evaluate that is this time fitting an encoding model. So we're not trying to predict the stimulus feature anymore. Now we're going to try and predict the neural response. So does a neuron spike, yes or no, based on the features that we've annotated of the sentences that people are listening to. So for this particular model, we take all of the phonemes across the English language, and we try to predict whether a neuron will fire based on what phoneme it is that just happened. And we can do that at relative lags to look at the time, the peak time of this neuron relative to when the phoneme happened. So this is the neuron that we were just listening to, and this is the weights of the TRF model. And what you can see is that the weights are fluctuating around 0, other than for the N and M sounds, which through our intuition from looking at those sentences, you will also can appreciate. So it seems that this neuron seems to be a nasal neuron. And we also find other neurons that encode similar types of phonetic features. So we have some that encode high vowels or low vowels, or fricatives, nasals, and approximants. So this was exciting to me because it's all of the examples of neurons that we found that seem to encode speech sounds did so with a phonetic feature code rather than a single neuron, rather than a single phoneme code. So we found lots of neurons that would fire to both M and N, but none that would fire to M in isolation from N, or vice versa. So it seems that when you go all the way down to the single cell, what the single cell cares about is the phonetic feature of a sound, and we didn't find any evidence for encoding of a phoneme category. So obviously speech is more than just the phonetic features. And so we did a more comprehensive annotation of these sentences too. So including phonetic features, but also fluctuations in the loudness of speech, the pitch of the person's voice, lexical stress, and then features relating to statistics. So how likely was this word to happen given the previous words, or how likely was this phoneme to happen given the previous phonemes. And so then that allows us to analyze all of the phonemes using all of these features and ask, okay, which neurons are best predicted by which of these features in our annotations? And how is that actually organized as a function of cortical depth? So on the right-hand side here, we have a plot that the y-axis corresponds to physical depth relative to the cortical surface. So here is the surface, and then we're going towards white matter over here, spanning all of the cortical layers. Each dot corresponds to one neuron, and the color of that dot tells you which of these features on the left-hand side best predicted whether that neuron was going to fire or not. And the x-axis tells you how reliable the model was. So how good were we at predicting whether the neuron was going to fire? So there's a few things I want to highlight here. One is that there are quite a lot of orange neurons and quite a lot of purple ones. So there's a lot of representation of encoding of pitch and encoding of acoustic phonetics. Relatively less encoding of envelope, but we still see a few. And we didn't find any examples of statistical features, either at the lexical or phoneme level. The second thing is that you can clearly see that this is not just randomly scattered around. We see that on average, the neurons which encode pitch tend to be encoded at a more superficial layer than the neurons which encode acoustic phonetic features. And this actually makes some sense, given what we understand about the kind of circuitry of neurons in these layers. So layer 4-5, that's receiving inputs from the thalamus directly, whereas in layer 2-3, these neurons have more recurrent connections, so they're more connected to each other, allowing you to do more longer timescale computations that might be necessary for something like pitch. So it seems that the where these neurons fall relative to the layers makes sense based on what we understand of the anatomy of what the inputs are to each of these layers. Okay, so to summarize this last study, when we're able to record from neurons across the cortical depth, what we find is that not all the neurons are tuned to the same thing. We see highly heterogeneous tuning. Even within one cortical column of one person, you find encoding of lots of different features in these neurons. And second, the tuning that we see has a depth-wise organization. So rather than a kind of salt and pepper, everything everywhere, we see that systematically certain features seem to be encoding a certain depths, and we need to, and that seems to make sense given kind of the canonical circuit of what we understand of where the inputs to these neurons are actually coming from. And I also want to give the broader take-home point of this work that this is super exciting that we're now able to actually record single neurons in humans at this scale. And this opens up a whole host of questions that we're now going to be able to address to understand speech processing from a totally new type of resolution. Okay, so to wrap up everything, when we're trying to understand this processing architecture of representations, operations, information flow, we see evidence of a very rich set of hierarchical representations generated, which we saw in the first study when listening to continuous speech have a top-down, then parallel, then bottom-up set of processes. We also saw that the processing of these features seems to unfold using a dynamic code. We did a deep dive showing you that for phonetics, but then also a quick snapshot of how that same code does seem to extend to the processing across the whole hierarchy. And we talked about how this enables longevity of encoding without interference between neighbors, and also gives an implicit code for the relative order of inputs in a sequence. And then finally, kind of a broader thing that I feel like I just find in every study that we do, it's just like how much parallel processing is going on here. So we saw parallel processing across the hierarchy. We also saw parallel processing in terms of the recent history of speech sounds. And then with the NeuroPixels Pro, we also see parallel processing of multiple features spatially within the same cortical column. And so this seems like a really important hint to me that parallel processing is a really important aspect of speech processing. Enabling very rapid communication between different formats of representation, between the future and the past, and between different neurons encoding different fundamental features. So with that, I want to thank you again so much for your attention, and I'd be very happy to take any questions.
Laura, thank you. Tremendous. I don't know if you can hear the applause in the room, maybe the acclaim on Zoom as well, but thank you so much. And I'll add, because I've been on the receiving end of this, when you're coming in on Zoom, the technical stuff has been perfect. We've heard the sounds, we've seen the slides look beautiful, so no tech glitches. Very, very clean presentation. Thank you. And we have plenty of time for questions. By my clock, we have just, we have plenty of time. So in the room, you can raise your hands. On Zoom, you can use the Q&A, and we will bring that question to attention. And you better bring your questions, otherwise you're going to hear from me and my questions, and no one wants that. and folks are getting the microphone to ask their questions. Thank you.
Great.
Yes, thank you very much. My name is Ryan Budnick. I very much enjoyed your talk. I have a question about experiment 2. We know that there are going to be strong correlations between the identities of adjacent phonemes because of phonotactics and because certain words are going to be more common than others. And so when we have one of these decoders that's able to identify, say, the identity of the previous phoneme, are we able to tell apart the extent to which it's capturing the continued neurological processing of that previous phoneme versus it's processing the current phoneme, and because there are correlations between phonemes, it's using that current information in order to make predictions about the previous one?
Yeah, thanks a lot for the question. So if what we were seeing was kind of an extension of the processing of the previous one, then we wouldn't see those neat diagonals like we do, because the information would be encoded in the same way as it was for the previous one as it is for the next. And so you would see kind of like a smearing of one into the other. On the note of prediction, though, we do see some interesting components of that. Let me pull that up quickly. Oh, okay. So this is phoneme 1 going forwards, and this is the last phoneme of the previous word going backwards. And there's a couple things. So one is that systematically the processing of the first phoneme happens later than the processing of the last one. And we thought, okay, maybe that's like a word boundary effect. But actually, when we analyze it for like word boundaries, so like word position, word position of the phoneme versus predictability, we find that predictability is a much better explainer of this temporal difference. So for sounds that you can expect, the processing starts earlier, and for sounds that you could less expect, the processing starts slightly later. So that's one. And then the second thing, which is related to prediction, is entropy, of how certain are you about what is about to happen. And we find that that is the best explanation of the longevity of the phonetic processing. So again, you can see it most strongly for the first versus last, but again, we modeled it using both that as a predictor and entropy as a predictor. And we find that if you are not so certain about what you're about to hear, you actually hold on to that phonetic information longer. Whereas if you do know what you're going to hear, you don't process the phonetics quite so long, which is super cool and interesting to me, because it suggests that there's some flexibility in the system, that you're basically holding on to the information for as long as you think you need to in order to resolve information at the level above. And this effect in particular, I don't have the plot here, is something that Jill was able to replicate using EEG, and that was actually one of the really critical distinctions between the aphasic group and the control group, is that the control group had this additional flexibility of the processing time and the aphasia group did not. So I'm super interested in what that tells us about like binding information together.
Do we have Zoom questions?
Okay, we have a question from Zoom that we're going to read out. Is that right?
Yes, I think we'll test.
Yeah.
So we have two questions from Zoom. So first one is from Huteng Dai, who is a assistant professor in our own department of linguistics.
Hello.
So Huteng says that, thank you, Laura. Do you think the position invariant feature code you found in perception has parallel in production, especially in how the system maps underlying representations to acoustic signals in speech? And that is exactly the question I want to ask because I am selfishly interested in production. So.
Yeah, no, that's a great question. I have not tested that myself. I'm trying to think of yeah, so I have collaborators building brain-computer interface devices for like production. I'm trying to think if I can think of any of their work that I can draw from, but I don't know that I can. My expectation would be yes, but I would love to test that directly. I can imagine that there is a part of the representation that is specific and a part of the representation that is invariant to position. And it'd be interesting to see, like, are we talking about motor cortex representations here? Probably. In which case, yeah, maybe the difference would be more in like how the sound needs to be articulated differently depending on the like yeah, like if this depending on what sounds are next to it, maybe it'll be voiced more or devoiced. And I imagine like the extent to which you see position differences, I can imagine it'd be more driven by those type of articulatory differences rather than a like fundamentally different code depending on where the sound is in the word.
To piggyback on that, Laura, can I ask, the BCI work you're referencing, is that work almost entirely based on recordings from motor cortex compared to these temporal or more higher order association areas? Or is there BCI production work that also covers some of these association areas?
Yeah, we're actually looking now at BCI like implants in inferior frontal gyrus, looking at much higher level phenomenon that we're looking at here, across actually both comprehension and production. So I know that's not the question that you asked, but that's also something that I find really fascinating is, okay, there's the question of invariance across position within comprehension or within production, but what about invariance across the production and comprehension systems? And so, yeah, in collaboration with them, we're also looking for that type of code too.
Thank you, Laura. And I'm wondering if I can like follow up with another production-related question before I move on to the next Zoom question. So, which is, since you mentioned about the two-way hierarchical representations in, so that's from natural listening, right? And I'm wondering if that's also the case in production, where you might also have like a prolonged, like consistent representation from like at the semantic vector level, and then you move on to syntactic operations, and then you move down at the bottom to like assemble the phonemes, and then you move up again. Like is that also manifested in production? Do you predict that?
Yeah. So I, again, haven't studied that in production yet. I do kind of feel like for production, the intuitive order of these features already is the like reverse. Like you come up with an idea of something that you want to say, and then you figure out like what sentence structure you need to put it in, and you figure out what words you should put in there, and then you actually say the sounds. So I would imagine that for production, we would also see this reverse ordering. And I don't think that prediction would move this around in the same way that it does for comprehension. So just to flesh that last point out a bit. So like, let's say that people weren't listening to continuous speech. Let's say that I just gave them like a short phrase. Then they have to be bottom up in processing that because there was no prior context upon which to bootstrap from. So in the case where it's just like an isolated phrase, then this would look very different. And that's another thing that we're actually working on right now, is to see how this shift in ordering actually changes as a function of prediction. But my very strong hypothesis is that if you're just hearing an isolated phrase, then it won't look like this reverse anymore. It's going to be from the low-level stuff up to the high-level stuff, because there's kind of no other option. And it's only when you have the context that you can flip it to be the reverse. However, if someone is talking, I think it doesn't really matter if they're saying a phrase or if they're saying an entire, I don't know, 50-minute presentation. I think it's going to need to work semantic down. Though maybe the relative time expands, that like you start being able to plan things much more in the future, and so the semantic starts being coded kind of earlier and earlier. But I don't think it would ever flip to the reverse, that you're planning you're processing the sounds you're going to say before you're processing the meaning of the sounds you're going to say. But I haven't done it.
Thank you, Laura. And is there
Let's go to the
more?
Zoom question and then another one in room.
Okay, so we have two more Zoom
Go to the one of the Zoom questions and then go to the
So
one
yeah,
in
so
room.
we'll get to the next Zoom question from Isaac Morin Martinez, who says, beautiful talk. You described how the brain carries different codes depending on acoustic or semantic features. I'm wondering whether you have hypothesized how the brain integrates information across the different levels in the hierarchy.
Yeah. I think maybe I deleted this slide. So yeah, so one study that I did, actually it focused in on word class to try to answer this question. So word class is convenient for two reasons. One, it's kind of in the middle of this hierarchy as I'm conveying it here. And two, words are often like super ambiguous as to their word class. Like one word form can be used for many word classes. So you can have duck can be a noun or a verb, for example. And so to try and look at how information gets passed between levels or how it gets integrated, we took word class and we tested whether we can better predict the neural response of word class based on the word form information. So in other words, what is the most frequent word class that duck is used for? Okay, duck most of the time is a noun, and so we're going to have the prediction that the brain will respond noun. The counter model is, no, word class is derived top down because it matters what the context is. And so the neural representation of word class, it won't be dependent on word form. It's going to be dependent on the syntactic structure. And so we also coded word class in a second way, which is what is the context sensitive label for word class in this sentence. And then I basically tested which of the two better predicted neural responses, and it was the contextual model, which suggests, again, that it's the high-level information being used to kind of derive the low-level representations. And that's kind of how information is integrated and used across the levels.
Let's go to a question in the room.
Thank you so much for that talk. It's really staggeringly impressive work, Martin Mosmer. My question was about intonation and more broadly tone. So first of all, let me try and remember my question. I've been waiting for a while. I'll remember the first part second. So I'll start with the second part of my question, which was, do you think that the interconnectedness of the neurons, so this is in the last study you talked about, in the part of the gray matter that looks at intonation tells us something about the complexity of that information and how it is constantly being mapped onto either the phonetics or other parts of meaning, or well, and do you think that in a language that is tonal, there would be even more activation of these pitch sensitive neurons? And do you anticipate that this would be different in a language with lexical intonation or tone versus tone on a syntactic level, so phrase level tone? I'll stop
Yeah,
there.
thanks for the question. So one thing that maybe is unsatisfying about what we have so far, but hopefully this will change in the future, is we don't really find too much kind of communication between the neurons that we're recording from. I was hoping that we'd really be able to like figure out a circuit where you're like, okay, yeah, the acoustic phonetics is like derived first, and then you kind of like assemble those to derive something higher order. But as far as what we have here, we don't have like too much kind of talking between the neurons for me to say too much about that. But yeah, hopefully, like and I would love to be able to understand like, okay, how does this stuff kind of come together to give rise to something like greater than itself? But I guess at the like more theoretical level, I do think it's interesting that we have relative pitch here. We also modeled absolute pitch, and that was a much worse predictor than relative pitch, which is interesting because relative pitch, you need to obviously do a normalization. So relative pitch means it doesn't matter if you have a low voice and I have a higher voice. What matters is, am I at the high part of my own range? And are you at the high part of your own range? Which is a pretty, it's like sophisticated like feature that is more than just what you could read out from the cochlea, for example. And so I think that like you asked about complexity, I think that being able to set up different representational spaces in that way and being like, okay, well, it's not absolute pitch, it seems like it's relative pitch, does tell you something about the complexity above what the alternative representational space could have been. I don't remember. I think, oh yeah, right, tonal languages. Yes. Yeah, I would love to look at that. All of the speakers here were native English speakers and did not speak any tonal languages. There has been work not at the single neuron level, but at the intracranial EEG level, looking at speakers of tonal and non-tonal languages. And indeed, the pitch features are used for like very different purposes and seems to be processed differently for tonal and non-tonal speakers. And that for sure would be also reflected at the single cell level. I do really wonder if like your pitch neurons kind of get bifurcated or something, that like some of the pitch neurons are doing like relative sentence pitch processing and other pitch neurons are doing like lexical identity pitch processing. That would be really fascinating. Something to do in the future.
All right.
Thanks, Laura. Can I insert myself in as a question? I'm going to do that. I'm just going to do it. Laura, and then we'll go to Zoom. Is that right? Okay. Yeah. Hi. So my question is about, it builds on this idea of actually what you were just saying about pitch neurons, but then I would love to hear your thoughts on connecting one of the main takeaways from experiment 2 and this takeaway from the single neuron work, right? So a main takeaway from experiment 2 was this idea of a dynamic code. And how are you squaring a dynamic code with this idea that there are, single unit receptive fields that are part of the key code for these abstract features?
Yeah, totally. Yeah, two answers. And this also came up a little bit in one of my meetings earlier. So for most of the neurons that we see, the receptive of neurons that code phonetic features, they look kind of like this. That the phonetic feature like 100 milliseconds before is what predicts the neuron will fire, and it's like very narrow in time. So what would the concrete prediction be under the dynamic coding hypothesis? It would be that you have redundancy of neuron, like a ton of neurons that like nasals, but some of the neurons like nasals 100 milliseconds ago, and other neurons like nasals 200 milliseconds ago, and others 300 milliseconds ago, etc. And so when you're listening to that whole population, you have this sequence where different neurons fire for the same feature, but at a different lag, and that's what gives you that dynamic code. And for the most part, we see receptive fields that look like this. There are, and what I mean is, and we don't find examples of neurons that fire way after the fact. We do find some little examples, like there were two nasal neurons in the recording of one patient where one of the nasal neurons peaked at 100 milliseconds and the other one peaked at 120. So maybe that is actually reflecting what I just described, and we just kind of got unlucky that we weren't able to, obviously we're vastly undersampling like all of the neurons in someone's brain. So maybe that is suggestive that what I described is true. Or the other thing, which I think is also possible, and we're starting to see maybe some evidence for with intracranial EEG, is that the code starts focal. And what I mean by that is you have a very large response from a small number of neurons, and then it becomes distributed and weak. So then you have way more neurons participating, let's say 100 times more, but any single one of those neurons is responding really weakly. So if you listen to just one of them, let's say this one, you would only be able to tell its strong response at 100 milliseconds, and you wouldn't be able to see that it's maybe also responding at like a longer lag. But if you're listening to the full population, what is common across the tons of neurons is the longer latency stuff. And what's specific to a given neuron is the short latency stuff. And so that's my kind of working hypothesis right now that we're trying to figure out. Like, are you, and exactly to your point, like, how do we reconcile these two results? And I think that, yeah, it's this shift from a focal code, which allows you to see things at a single neuron level, to a distributed code where there you really need to listen to the population to be able to decode the information from it.
Yeah,
I hope that makes sense.
Exciting, exciting. We have time for one more question.
So the next Zoom question is coming from Yike Li, who is a PhD student in psychology department here in Michigan. So she says that, "Hi Laura, great talk. Would the order of processing hierarchy change if looking at audio-visual speech, since visual speech information might influence the predictability of phoneme and subphoneme features more?"
Cool idea. Again, haven't do that. I'm realizing just how much work I've got to do. But that's a really interesting idea. I agree with you. I think that that would change the dynamics, and I agree. I think that if anything, it would just kind of push these processes earlier. But yeah, that would be super interesting to look at productivity based on the visual stream and helping to process the auditory speech stream, as well as prediction just kind of within the speech stream itself, and see how these different dynamics move around depending on the predictive information available. Thanks for the question.
Great, thank you, Laura. These are excellent questions and also excellent answers. So, any
Yeah.
other questions from in person? I think we still have like.
I think we are out of time, but this is sad for us, Laura. Sad for us to say goodbye. But this has been great. This has been a great conversation, great presentation. Thank you for joining us today. We really appreciate it. Sorry you couldn't enjoy actually a very, again, pleasant Ann Arbor November day. I hope someone got outside today, but I'm sure it was nice in Palo Alto as well. And well, thank you so much.
Great. Thank you so much. I've really enjoyed today. Thank you. Hope to see many of you soon.
And I don't know what happens on Zoom at this point, but it's going to unceremoniously end.
Yeah.
Have a good afternoon. Bye. Thank you all for coming today. Grab snacks. All right. Thank you, Danielle, thank you.